May God's peace be on all of you. Our today's lecture deals with essential components of fermentation media. Water. All fermentation processes, except solid substrate fermentations, require vast quantities of water. In many cases it also provides trace mineral elements. Not only is water a major component of all media, but it is important for ancillary equipment and cleaning. A reliable source of large quantities of clean water, of consistent composition, is therefore essential. Diffuse removal of suspended solids, colloids and microorganisms is usually required. When the water supply is hard, it is treated to remove salts such as calcium carbonate. Iron and chlorine may also require removal. For some fermentations, notably plant and animal cell culture, the water must be highly purified. Minerals. Normally, sufficient quantities of cobalt, copper, iron, manganese, molybdenum, and zinc are present in the water supplies, and as impurities in other media ingredients. For example, corn steep liquor contains a wide range of minerals that will usually satisfy the minor and trace mineral needs. Occasionally, levels of calcium, magnesium, phosphorus, potassium, sulfur and chloride ions are too low to fulfill requirements, and these may be added as specific salts. Vitamins and growth factors. Many bacteria can synthesize all necessary vitamins from basic elements. For other bacteria, filamentous fungi and yeasts, they must be added as supplements to the fermentation medium. Most natural carbon and nitrogen sources also contain at least some of the required vitamins as minor contaminants. Other necessary growth factors, amino acids, nucleotides, fatty acids and sterols, are added either in pure form or, for economic reasons, as less expensive plant and animal extracts. Precursors. Some fermentations must be supplemented with specific precursors, notably for secondary metabolite production. When required, they are often added in controlled quantities and in a relatively pure form. Examples include phenylacetic acid, or phenylestamide added as side chain precursors in penicillin production. D-threonine is used as a precursor in L-isoleucine production by Serratia marcescens. And anthranilic acid additions are made to fermentations of the yeast, Hansnula anomala, during L-tryptophan production. Inducers and elicitors. If a product formation is dependent upon the presence of a specific inducer compound, or a structural analog, it must be incorporated into the culture medium, or added at a specific point during the fermentation. In plant cell culture, the production of secondary metabolites, such as flavoids and terpenoids, can be triggered by adding elicitors. These may be isolated from various microorganisms, particularly plant pathogens. Inducers are often necessary in fermentations of genetically modified microorganisms. This is because the growth of GMMs can be impaired when the cloned genes are switched on, due to the very high levels of their transcription and translation. Consequently, inducible systems for the cloned genes are incorporated, that allow initial maximization of growth to establish high biomass density, whereupon the cloned gene can then be switched on by the addition of the specific chemical inducer. Inhibitors. Inhibitors are used to redirect metabolism towards the target product and reduce formation of other metabolic intermediates. Others halt a pathway at a certain point to prevent further metabolism of the target product. An example of an inhibitor specifically employed to redirect metabolism is sodium bisulfite, which is used in the production of glycerol by Saccharomyces cerevisiae. Some GMMs contain plasmids bearing an antibiotic resistance gene, as well as the heterologous gene. The incorporation of this antibiotic into the medium used for the production of the heterologous product, selectively inhibits any plasmid-free cells that may arise. Cell permeability modifiers. These compounds increase cell permeability by modifying cell walls or membranes. promoting the release of intracellular products into the fermentation medium. Compounds used for this purpose include penicillins and surfactants. They are frequently added to amino acid fermentations, including processes for producing L-glutamic acid using members of the genera Corynebacterium and Brevibacterium. Oxygen. Depending on the amount of oxygen required by the organism, it may be supplied in the form of air containing about 21% oxygen, or occasionally as pure oxygen when requirements are particularly high. The organism's oxygen requirements may vary widely depending upon the carbon source. For most fermentations, the air or oxygen supply is filter sterilized prior to being injected into the fermenter. Antifoams. Antifoams are necessary to reduce foam formation during fermentation. Foaming is largely due to media proteins that become attached to the air broth interface where they denature to form a stable foam. If uncontrolled, the foam may block air filters, resulting in the loss of aseptic conditions. The fermenter becomes contaminated, and microorganisms are released into the environment. Of possibly most importance is the need to allow freeboard in fermenters, to provide space for the foam generated. If foaming is minimized, then throughputs can be increased. There are three possible approaches to controlling foam production: modification of medium composition, use of mechanical foam breakers, and addition of chemical antifoams. Chemical antifoams are surface active agents, which reduce the surface tension that binds the foam together. The ideal antifoam should have the following properties: readily and rapidly dispersed with rapid action, high activity at low concentrations, prolonged action, non-toxic to fermentation microorganisms, humans or animals, low cost, thermostability, and compatibility with other media components in the process, that is having no effect on oxygen transfer rates or downstream processing operations. Natural antifoams include plant oils, for example from soybean, sunflower and rapeseed, deodorized fish oil, mineral oils and tallow. The synthetic antifoams are mostly silicon oils, polyalcohols, and alkylated glycols. Some of these may adversely affect downstream processing steps, especially membrane filtration. Animal cell culture media. Animal cell culture media are normally based on complex basal media, such as Eagle's cell culture medium, which contains glucose, mineral salts, vitamins, and amino acids. For mammalian cells, a serum is usually added, such as fetal calf serum, newborn calf serum or horse serum. Plant cell culture media. In contrast to most animal cell culture media, those used for plant cell culture are usually chemically defined. They contain an organic carbon source, as most plant cells are grown heterotrophically, a nitrogen source, mineral salts, and growth hormones. The combination and concentration of plant hormones provided depend upon the specific fermentation. Auxins are usually supplied, along with cytokinins to promote cell division. That's it for today's lecture. Don't forget to subscribe, and we will see you guys next time.